Thanks, Anil. Thanks everybody for joining today. It's 10 o'clock, so let's get going. Today I'll be presenting a variety of measurements that show how the Spectradyne's ARC particle analyzer can be used to characterize LNPs. And some of those will be in just illustrative examples, and some will be in much more complex samples in doing experiments that can't really be done in any other way. So I look forward to hearing your questions. And as Inal said, please feel free to enter them in the chat at any time during the meeting. We'll talk about them at the end together. So if you're not familiar with Spectradyne, we make breakthrough instrumentation for accurate nanoparticle analysis. And our goal is to reshape the discovery, development, and production of modern therapeutics. And those take a lot of different forms. But specifically, the three critical quality attributes for nanomedicines that we characterize are these ones. So particle size, this is critical because it impacts directly the biodistribution of nanomedicines. It also determines the dose because it determines the size of the particles that can be delivered, as well as their stability in formulation. The concentration of these particles, obviously, that's related to the dose, and the concentration of the different components, including empty and full LNPs, for example, relate to the toxicity of the formulation overall. And the incorporated payload into the particles also important because of dose and efficacy. Ultimately, you want to make sure that your drug is getting into the particles so that it can be delivered properly. All these are critical quality attributes that add up to our goal, which is safe and effective therapeutics. And I'd like to illustrate the importance of these three CQAs in a canonical example. So in this example, what we're trying to do is compare the potency of two formulations, A and B. Okay. So the way this is typically done is with the formulations are applied to a test system. Here we're illustrating this with a tissue culture example. And formulation A turns some of the cells fluorescent, and formulation B turns a lot more of them fluorescent. And we measure that using some readout like flow cytometry maybe for the cells to determine the number of fluorescent cells that came out. So this is all well and good. We see a difference, right? But it's very important when you're doing this experiment to first control for the dose, the amount of material that you're putting in, right? If you put in 10 times as much of formulation B, then obviously you expect to see a different response in the test system, right? So it's very important for basic good science to be able to measure the size, concentration, and the payload of these nanomedicines in order to do good experiments, right? So current ways for doing this are diverse, right? There are many different technologies out there for attempting to measure nanoparticles, characterize them, and you probably use a mix or maybe all of these methods. So typically there's a dynamic light scattering instrument in the LNP lab, right? In your LNP lab. And so this is an averaging method that reports an average scattering intensity diameter. Not a real particle size distribution. Then for size and concentration, two main workhorses are NTA for nanoparticle or nanoparticle tracking analysis, and they're increasingly nanoscale flow cytometer instruments being developed for nanoscale particle analysis, right? And both of these use light scattering to detect the particles. And then that's for size and concentration. And then for payload, really, there's only bulk methods available. So these are like the qubit assay, the ribogreen assay, and these tell you the total amount of RNA that is encapsulated in the particles, but it's an average. It doesn't tell you the distribution of encapsulation, right, or how much of your particles are loaded versus empty, for example, or the degree of loading. So there are a couple of, in addition to that, there are a couple of commonalities worth looking at across this board. So all of these techniques use optics to measure the size and concentration of the particles, okay? And so what that means is that you're looking from just one perspective, which is the light scattering perspective, to measure these particles. That's an indirect method for measuring the particle size, and so that means you need to do complex data interpretation to infer particle size, and I'll show you some examples of that in a minute. It also means that if you're measuring a heterogeneous sample, so for example, in particular for size measurements with light scattering, if you have a heterogeneous sample, you can't make the right assumptions about the particles that are accurate for all the particles in the sample, particularly the index of refraction. And I'll show you an example of that in a second to illustrate that. So as soon as you have a heterogeneous sample, the size and concentration distributions that these instruments are reporting are not accurate. And so I'd like to just dive into that for a second for the flow cytometry application, for example, because a lot of people are trying to use this, and it's important to understand how it works and how it doesn't work. So this plot is taken from a paper in Scientific Reports in 2019 from Beckman-Coulter. And this is a really nice paper. What they did was they measured on a flow cytometer a bunch of different materials of different sizes, okay? And that's shown here. So the green dots are all measurements of polystyrene beads of different sizes, known sizes. And then the yellow ones are viruses, right? And then the blue ones are silica particles, okay? So the point of this paper was to show that they could model the index of refraction of these different materials well, and that's what these gray lines show. So the lines are constant index of refraction, and you can see that the constant materials generally follow along these streamlines properly. And this plot is showing the scattering intensity on the y-axis, so how much the particles scatter light versus their actual particle diameter, right, as measured by some other technique. So what I'd like to do is imagine mixing the three of these materials together. So we've got 125 nanometer polystyrene, we've got HSV1, which is around 150 nanometers, and then a larger 200 nanometer silica particle, okay? And the reason we're doing this thought experiment is to simulate a complex mixture of particles with different refractive indices. And this could be just a single LNP formulation with a mixture of empty and loaded particles, right? Because the loading affects the refractive index of the particles. Okay, so the true particle size distribution of this sample looks something like this, right? The 125 nanometer polystyrene is the actual smallest particle in the distribution. So its peak is over here, HSV is in the middle, and silica is largest, right? 200 nanometer diameter. But if we try and infer the size of this distribution using light scattering alone, we get a different distribution, right? Because the index of refraction of the polystyrene is so much higher than the silica, it actually comes out as a larger inferred size than the silica. The silica is almost twice as big, right? And then the HSV comes out at the smallest inferred particle size because it scatters the least light. So its scattering intensity is the weakest. Now, if you had a pure sample of HSV, you could just, enter a different refractive index and to calibrate it, right? And then you would know that particle, then you could measure its size properly. But in a mixture, and particularly in samples where you don't know the composition in advance, like any real world sample, right? Then this size distribution that you infer from light scattering is going to be all messed up, all right? And so what we need is more accurate and more direct methods for measuring particle size and also concentration and payload, right? So we can do all that together. That's where Spectradyne comes in. Okay, so our product is the ARC particle analyzer, and it makes direct, accurate measurements of nanoparticles down to about 50 nanometers currently, and we're working to push that smaller. It measures size, concentration, and the payload on a particle-by-particle basis, and I'll show you examples of that. Very importantly is this technology does not require any assumptions about the sample in advance. So the ARC's accuracy is independent of the particle's refractive index, the polydispersity, so the mixture of different sizes, or the sample heterogeneity. So you can have a mixture of a bunch of different materials in there, and we'll still give you an accurate size and concentration distribution. It's also very fast and easy to use, especially compared to flow cytometry. There's no alignment required, there's no calibration required, and no cleaning required between samples. And the sample prep is very easy compared to flow cytometry. There's a single step staining usually, and no washing required of the sample before measurement. And I'll show you examples of all those things today. So let me spend a minute describing how it works. So the magic happens inside this microfluidic cartridge. And we use an electrical method that we call MRPS. It's microfluidic resistive pulse sensing to measure the size and concentration inside this microfluidic cartridge. What we do is we flow the particles through the bottleneck in this constriction, or this constriction in the fluid flow, okay, the bottleneck, and particles flow through one by one. Every time a single particle flows through the constriction, we get an electrical signal that tells us how big the particle is and how fast it went through, okay? And the speed tells us the flow rate of the sample that's flowing through the constriction. That allows us to calculate the volume of the sample that we measured. So we're counting particles in a known volume. That's the most direct method of concentration that you can get. We're not inferring the particle size from any light scattering or anything. It's really just directly measuring the physical size of the particle as it passes through the constriction. That's how this RPS technology works. It's been around for decades in terms of the Coulter counter, right? Now, in the arc particle analyzer, what we're doing is something totally new. We're exciting with a laser beam every particle simultaneously with the electrical measurement and measuring the fluorescence light that that particle emits at the same time as we measure its size electrically, okay? So this is how we measure fluorescence or surface markers on different kinds of particles, or the internal payload, or membrane dyes. There's a lot we can do with this technology, just like on a flow cytometer, but now at the nanoscale properly. One of the reasons this works is because of Spectradyne's patented microfluidics. These are highly engineered cartridges that include features that allow you to measure very complex samples in the cartridge. So one thing I'm showing up here in the far top right, panel A, is an embedded input filter that's embedded in each cartridge, so it excludes large particles that might otherwise block this constriction as it comes through, okay? So ultimately, all this engineering equates to ease of use for you, the end user. It allows you to measure complex samples. So I'll show you examples of nanoparticles in plasma today, other biofluids or tissue culture media. These cartridges enable the pre-calibration, so you don't have to do any calibration or alignment also of your instrument or cartridges, and affords very small sample volume requirements. So just 3 microliters is all that's needed. So let me show you just an illustrative example to show you how the technology works. Okay, so here we mix up a giant mix of 6 different polystyrene beads with different fluorochromes. So this is the component, these are the components of the mixture. Okay, we have some non-fluorescent particles, we have some overlapping but different fluorochrome particles, and different sizes, right? All spanning from about 100 to 300 nanometers this sample. And we mix them together equal concentrations. This is what the raw data looks like. So what we're showing here are, in the blue is the electrical signals. So these are detecting individual particles, right? One by one as they pass through the constriction. And you can see this first particle is fluorescent and generates simultaneous fluorescent signals in each of our fluorescent channels, detection channels. Whereas this second particle that comes through is a non-fluorescent particle. And so it doesn't generate any fluorescent signal in those channels, all right? So when we look at this sample as a whole, this is the kind of data that we report. So this is each one of these curves shows the concentration on the y-axis versus true particle diameter. So this is measured electrically, right, with the MRPS method, for different populations in this mixture. So in blue is the distribution of all the particles that are detected by MRPS. And you can see it's got many different humps, right? In green is the distribution of just the green fluorescent particles, and most of these are also fluorescent, except for these 100 and 200s, which are non-fluorescent, right? Et cetera, through the three different channels, okay? So what you can see is that in terms of size, we can resolve all these different sizes, right? But this 180 nanometer peak is still composed of two different particles that have overlapping size distributions, right? So let's look at the fluorescence information. Now, in this data, we'll be seeing a lot of this data today. In the y-axis, we're showing the brightness in our green channel, the FL1 channel. And then the other two panels are yellow and our red channel, right? So using these other dimensions of fluorescence, we can then we can isolate and identify all of these different populations of particles in the mixture. The fluorescent, the non-fluorescent particles, the 100 and the 200 nanometer particles, are below our limit of detection, right? They're not fluorescent. The red ones are here, and they're the only ones that show up in our red channel. And we can now distinguish between these two different fluorochromes on same size particles based on their brightness in the green and the yellow channels. Okay, so this example just illustrates the kind of resolving power for quantifying subpopulations of particles in complex mixtures that you can do with the ARC. Let's get into more interesting examples. So Spectradyne focuses on three key areas: nanomedicine, in terms of primarily LNPs and synthetic particles, virus and gene therapy applications, as well as extracellular vesicles. Today we're going to focus on LNPs, because that's what we said we do today. So this first example is going to show you, describe how you would use the ARC in a formulation development application. So we're going to compare three different RNA-loaded LNP formulations. The sample prep is very straightforward. We stain with a membrane-permeable intercalating dye that fluoresces in the green or FITC channel. Okay, so we'll focus on the green channel for now. It's a single-step staining, so there's no wash required afterwards. We stain for about 10 minutes and then measure the size and concentration and the fluorescent payload on the ARC in about 5 minutes per sample. Okay. These three formulations were measured also by dynamic light scattering, so we've included that measurement here for reference, right? Remember, DLS does not actually measure a size distribution. It does not measure the concentration, okay, unless you make assumptions about the particle's index of refraction, like we talked about before. So that doesn't work for the fluorescent or for the heterogeneous samples. And so really what we get are this Z average number for the three different formulations. And you can see what they are here. So there's a slight difference between these formulations from a DLS perspective this way. When we measure these particles on the ARC, these are the particle size distributions that we see. So this is just using the electrical information. So we're showing concentration versus particle diameter here for the three formulations. And you can see subtle differences between them. Formulation A has a hump of larger particles out here, whereas green and red are pretty, much closer similarity in terms of particle size distribution. We can be quantitative over any size range here, but over the full measured size range, we can report the concentration and the median particle diameter. So they're generally similar, but there are some important differences here. The story gets a lot more interesting when we look at the fluorescence, though. Okay, so now we're looking at a scatter plot that shows the brightness on the y-axis for every particle. Each dot represents a particle that's being detected, okay, versus true particle diameter measured on the ARC for these three different formulations. And here we can see very strongly that formulation C has a lot less incorporated payload than formulations B or A, right? So we have a detection limit on the instrument like any other technology. And so we distinguish between fluorescent positive and fluorescent negative samples here. And we'll look at a lot of data that looks like this today. And so we can say that the ones that are fluorescent are the loaded ones because they picked up the intercalating dye. and the ones below the line that are not fluorescent are empty, okay? And we've demonstrated this with empty LNP controls, for example. We can be quantitative over the fraction of loaded particles in these formulations. So the blue sample contains the highest fraction of loaded particles, about 43% of all the particles that are detected on this size range. Green, formulation B, comes in a little bit behind, and formulation C is really kind of a dud formulation, right? Only 3% of the particles are loaded with the payload in this example, right? We can take this a step further because our brightness measurements are quantitative, right? So we can look at the distribution of brightnesses in the sample and learn even more about the encapsulation process, or how the payload is encapsulated in these samples. So formulation C has the lowest concentration. So this is showing the concentration on the y-axis versus the brightness of the particles on the x-axis. These are both log scales, right? So what we see is that the formulation C has a much lower concentration of bright particles than formulation A or B, right? And there are some subtle differences between formulation A and B also. So formulation A has a much higher concentration of highly loaded particles than formulation B in green, whereas formulation B has a higher concentration of less loaded particles than formulation A. And we can be quantitative, again, reporting median brightness of these particles in this distribution over the loaded size range, right? Then we can put a gate just around the loaded particles, right, and look at their size distribution. So really, this is what you want as a formulation scientist. You want to know what's the size and concentration of the particles that are loaded with the payload, right? And so that's exactly what this is telling you. So in formulation A, we've got a slightly larger median particle size than formulation B, and these are just of the payload carrying particles, right? And again, formulation C is a smaller size of loaded particles and a much, much lower concentration of loaded particles in that formulation. All right. So this example shows that the ARC can directly measure RNA payload on a single particle basis. So compare this to the information that you get from a ribogreen assay, for example, which just tells you that you've loaded so much RNA into the LNPs. We're getting a much richer information set from this analysis. Just takes 5 minutes. It's actually much simpler than doing the ribogreen assay. We've quantified the loading efficiency in terms of full and empty as a function of particle size. So that's what we're talking about in comparison to ribogreen. We can measure the concentration and size of the loaded and the empty LNPs separately, right? Because the empty LNPs are also important for toxicity and impedance of uptake reasons, right? And all this adds up to allow you to evaluate your formulations faster and more directly using the ARC rather than the DLS combined with ribogreen assay. Okay, so remember, opportunity is to type in your questions at any time in the chat, and we can talk about them at the end. So you may ask, how do we know how well this is working, right? So let me show you a few examples of validation experiments that we can do on our ARC because it's such a direct measurement, right? So first of all, we can ask about the repeatability of the staining and the measurement. So we took one RNA-loaded LNP sample. We measured it in triplicate on separate days, right? So we stain each replicate. So each day we stain it, and then we measure the size and concentration on the ARC, and we can look at the size and loading and everything we've talked about already. So these are those 3 different replicates. We're getting very good agreement between, so this is showing the brightness on the y-axis again versus particle diameter on the x-axis. And you can see very good repeatability between the brightness distribution here from day to day and from staining replicate to staining replicate. This sample is really interesting, right? Because there's a very clear difference between empty and full here, right? You can tell at 200 nanometers, these particles are either full, right, or they're empty, and there's not a whole lot of gray area in between here, right? Not only do these sort of clouds overlap very well, but because our fluorescence is quantitative, right, we can look at how well the distribution of brightnesses of particles in these samples agree. Very highly repeatable over, many orders of magnitude in both directions, right? Concentration versus brightness. We have excellent agreement between replicates. Okay. So another question you might ask is, how do you know that you're staining the payload, right? And so let's prove that to you. And this is an example this is something that only we can do on the ARC because of the direct measurement of the size and concentration. So what we're going to ask is, how does the brightness depend on size of the particle? If we're staining something like the surface of the LNPs and not the internal payload that we're looking for, then we would expect a different scaling of brightness on size than if we're really staining the inside, right? That scales with the volume. So same sample prep, we stain with this intercalating dye, no wash steps, takes about 10 minutes, and then we measure them on the ARC. And then this is the distribution that we see. Okay, so similar to the last example, we get this sort of curving in these log linear plots, curving distribution of brightness. So let's ask, how does the brightness depend on particle diameter? So if we look at that on a log-log plot like this, we can see that the brightness depends on the third power of the diameter. Okay, and so without going into the math, right, the brightness is proportional to the volume of the particle, not its surface area, right? So what we're showing here is that we're staining the internal payload, right? The internal volume of the particles. That's where the RNA lives. It's not something on the surface that is picking up the stain, right? So this shows that we're doing internal staining of the internal payload, and also that the particles are remaining intact through this measurement, because we would not see this volume scaling if the particles were disintegrating, for example. All right, so we've shown you the validation stuff. Let's get into more interesting measurements in terms of complex matrices. Okay, so this next example is going to be a simulation of a PK testing, experiment, where we simulate a test of measuring the fate of LNPs or any other nanomedicine in plasma, and we're going to measure them directly in the biofluid, right in the plasma. Okay, so what we did here was we took fluorescent particles, we serially diluted them into cell-free plasma. Okay, and in each of those samples, we measure the size concentration of the whole sample, as well as the subpopulation of fluorescent particles. Okay. This is just one of those samples, so we can look at it in detail. So these are 100 nanometer fluorescent particles, right? Here they are. Okay, and this is showing concentration versus particle diameter for two different populations of particles in the sample. So in blue are all the particles, so that's a mixture of the fluorescent particles and the plasma particles, right? And in green, or I'm sorry, the blue is a mixture of the plasma particles and the fluorescent particles. And then the green is just the concentration, the particle size distribution of the fluorescent subpopulation. So these are the particles that are bright enough to be detected above threshold, right? And so what you can see here is that at this high spike in concentration, the fluorescent particles actually just cause a bump in the overall particle size distribution, right? They're high enough that they stick out over the background of particles in the plasma. But as we serially dilute this fluorescent subpopulation, right, you can see that their concentration goes down until they're indistinguishable from the overall particle size distribution, right? So this bump on top decreases until the concentration of the fluorescent particles is well below the concentration of plasma particles in there that are at a constant concentration, right, in each of these samples. So we can look at this data in a number of different ways. This shows all the different, the total particle size distribution of all the samples, and you can see this bump just kind of disappears at a low enough spike in concentration. This is showing the brightness scatter plot like we've been looking at, and for each of the samples. And you can see that the fluorescent subpopulation at 100 nanometers sticks out like a sore thumb in every one of them, right? It's very easy to identify these particles from the plasma background based on the fluorescence. If we gate to just the fluorescent subpopulation, then these are the distributions that we get. And if we measure the concentration as a function of dilution factor for the fluorescent population, we get a very good linear response, all the way down to almost 100-fold lower concentration than the plasma particles themselves, right? So here the spike in is at 100-fold lower concentration than the plasma, and we're still able to detect them accurately in this sample, right? So this simulation of a PK testing, so in a PK test, just to go back here, you would spike in LNPs at the beginning into the plasma, and you would watch them evolve over time instead of at different concentrations, right? And that tells you the stability and the fate of those nanoparticles, the intact particles, directly in the biofluid over time there, right? And so that's what this example is meant to illustrate, to show that we can do this accurately for fluorescent particles in such a complex mixture that way. Okay, so now let's do it for real. So let's use LNPs and let's spike them into serum. Okay, so in this example, what we did was we mixed LNPs into serum, or we used a PBS control, so we'll show that also. We stain the LNPs directly in the plasma mixture, okay? And then we, at every time point, okay? And then we stain, and then we sample them over time, okay? So at each time point, take the particles out, stain them, and then measure them on the arc. First of all, in PBS, what we see is there's essentially no degradation over time, right? So across an hour and a half, we're seeing very consistent fluorescent distributions and concentrations of these LNPs over time, right? When we do this experiment in serum, we see something qualitatively and quantitatively different than in PBS, right? So there's an immediate decrease in the concentration of the LNPs at our shortest time point, and then there's a decrease over time where the concentration of fluorescent particles is decreasing as these particles degrade over time. And we can be quantitative about this, look at the concentration of fluorescent particles of the LNPs in serum. Those are the dots in blue. They're relatively constant over the time of the measurement, and compare that to what's done in the serum in red, okay? And you can see there's a decrease and a decay form over here. So I hope that has spurred some thoughts for how you might be able to use the ARC. So the ARC is really making very direct and very accurate measurements of LNP size, concentration, and payload at the nanoscale, down to about 50 nanometers. There are no assumptions required about the sample in order to make this work. Its accuracy is independent of refractive index, polydispersity, or the sample heterogeneity, right? We've measured mixtures of polystyrene and LNPs and plasma particles all mixed together, and we get accurate size distributions of those complex samples. It's very fast and easy to use. We were able to run those samples directly on the chip, right? As well as what you don't see in the data is that there's no alignment process, no calibration required, or cleaning in between samples, even though we're doing these very complex matrices, right? If you would like to learn more, we encourage you to come to our website. We've got a lot of resources available there to learn about the technology, how it works, and application notes in LNPs, but also in our other application focus areas as well. And so with that, I'll say thank you very much and be happy to take any questions that you have.
Thanks very much, Dr. Frickin. Ladies and gentlemen, if you'd like to type in questions, you can do so in the Q&A box as you've done. We have 7 questions, which Dr. Frickin can walk through. Also, if you'd like to ask a question in real time, you're welcome to raise your hand electronically and we'll unmute you. So perhaps if I could ask Dr. Freikin to go through the questions in the order received and kindly address them. That would be great. Thank you,
Sure.
Dr.
Yep. Okay, so first question is from Regina, who is asking, hi Regina, by the way, so asking about, it says, the question is, our sample is polydispersed with particles below 65 nanometers, so but below the cutoff of the cartridges. So, and the question is, how does this affect the accuracy of the measurement? So the answer is that it does not affect the accuracy of the measurement. The reason there's a limit of detection for any given cartridge size is that particles that are below that level are too small to generate an appreciable signal that can be detected, right? And so they just pass through the constriction without generating a signal, and so that doesn't affect the measurement, right? Next question is from Luis Perez, thank you. Is hydrodynamic diameter what Spectradyne measures as true particle size? Okay, so this is a very good question, and the answer is no. We're not measuring hydrodynamic diameter. So hydrodynamic diameter is the diameter of a particle that diffuses in the way that's observed, right? And so a lot of things can affect the hydrodynamic diameter without appreciably actually changing the particle size. What we measure is just the core particle itself. So we typically measure close to what a TEM measurement would say, rather than what NTA or DLS measurement would say. Those ones measure hydrodynamic radius, right? So what we're measuring directly is the volume of the particle that excludes, that as it passes through the constriction. So we're measuring the physical size, physical volume of each particle as it goes through. Hope that answers your question. All right. Here is another question. How does the LNP loading quantification by ARC compare to other methods of quantifying methodologies? Thank you. Thanks for your question. So there was a recent paper from someone at Johns Hopkins who, so actually, let me go back. So typically we measure more empty particles than, say, you would on a nano FCM instrument. And the reason being, we think, is that the light scattering based instruments tend not to detect the weaker scattering particles, right? And so, but for us, we measure everything that's above our limit of detection. And so we quantify more accurately the full and the empty. And so if you're looking at a percentage basis, it looks different than what you would get from a light scattering based method, okay? In terms of fraction load-in, we measure similar to what you see in the literature, which was what I was trying to refer to before. So there's a recent paper. I don't think I can probably drop it into the Q&A right now, but if you follow up with us after by email, be happy to send you the reference. It's a paper from JHU recently looking at empty versus full using a purely optical technique. So they're not measuring size, but they have a way of measuring the combination of envelope and content, right? And so they measure somewhere between 40 to 60% empty LNPs in a standard formulation. And that's pretty consistent with what we measure also. All right. Next question. How would you determine the RNA encapsulation efficiency in the ARC? Okay. It seems like the analysis gives you the proportion of LNP loaded versus empty instead of the total RNA amount encapsulated. Okay, that's a very good question. So encapsulation efficiency is what you get from ribogreen assay, right? Which tells you the amount of your input RNA that gets encapsulated in your particles, right? And so that can be very high, even if it's only going into a few of the particles, right? So the ribogreen assay does not tell you the distribution of encapsulation, right? we measure the distribution of encapsulation, but we do not measure the free RNA in solution, right? So we're measuring something different than encapsulation efficiency as normally thought of in terms of the ribogreen assay. We measure the RNA, the loaded versus empty, and we are working on the calibration to tell you the number of micrograms of RNA per mil or per particle, but it's coming. Okay, so be patient, it's on the way. Can we measure fluorescence at multiple wavelengths simultaneously? Yes, we can. And that's what I showed you in the bead example at the beginning. We also have examples where we can co-localize different colors on single particles, right, with immunostaining or different stains together. That's all possible on the ARC, just like you would on a flow cytometer, for example, right? Okay, next question is about GMP compatible, right? Yes, we have a GMP package for the CFR 21 compliance that we can add on to the technology. Yes, and we'd be happy to work with you with the details of validation of your process using our technology, right? Let's see. Next question. Did you validate your PK analysis, so fluorescence particles, LNPs, EVs, and plasma, with standard quantification methods? Did you get a good correlation? So what I would ask is, what standard quantification method would you use to measure the intact concentration or the concentration of intact particles in the plasma? As far as I know, there's not a good way to do that. The way to do that is commonly used would be to measure the fluorescence in that plasma sample, but that doesn't actually tell you about the concentration of intact particles in that sample, right? Because as they break up, then the fluorescence still persists in a bulk assay format, right? Next question. What are the important parameters to ensure accurate size and particle measurement in the ARC? In other words, what are the source of errors in the ARC? Okay, so we've talked about the things that that do not impact the measurement, which are most things, right? So polydispersity does not impact the accuracy of the measurement. The heterogeneity of the sample does not impact the accuracy of the measurement. What does impact the accuracy of the measurement? Well, sizing depends on the conductivity of the sample, for example. So if you significantly change the concentration of salt in the sample, that can affect things. But what we recommend for that application is just to obtain the new calibration for that material by spiking in a particle of a known size. So it's very easy to do that on our system. You can't do that with other technologies, but you can always add a reference material to your sample that serves as a built-in positive control for the measurement, as well as an internal calibrator, right? And with fluorescence, you can exclude that very easily with gating and stuff. So you can add that to the sample and then not look at it again, but you can use that as a normalization for your data. All right. Next question. Could you summarize in a nutshell the greatest advantage of ARC with respect to NTA? Okay, I love this question. Thank you for asking it. NTA does not have, it's really not very quantitative. Okay, so I know people like to use it that way, but first of all, in terms of size, right, the limit of detection of NTA is dependent on the sample that you're trying to measure, right? And so a lot of the measurements of NTA show a nice peak around 100 nanometers and then a drop off at lower sizes, right? That's very commonly presented, but that peak is often an artifact of the measurement technique itself, okay? But what's happening is that the NTA instrument cannot detect the small particles in the presence of big ones. And you can do that you can prove that to yourself very easily if you're an NTA user. Just mix 2 or 3 different bead sizes together at equal concentrations, and you will see that the smallest ones disappear in the mixture, even though they can be detected in a monodispersed sample. And we have lots of literature like that on our website. We're happy to talk about that. So that's the greatest advantage would be of our technology is the accurate size and concentration of very complex polydispersed samples, right? When it comes to fluorescence for NTA, that's another whole can of worms. And the reason is because the limit of detection in the fluorescence dimension, like it's not quantitative for fluorescence, right? You don't know if you're not seeing a particle because it's just not bright enough or because it's not fluorescent. The ARC has very clear, very well-defined limits of detection in all its measurement dimensions. And so when you say the concentration of fluorescent particles is X, you can put bounds on that number in a way that other people can use to actually use the data, right? So it's really quantitative versus qualitative, honestly, the difference, right? Okay, next question, is the ARC able to detect number of RNA copies per LNP? So that's the part we're working on, is the conversion of brightness of a single particle to mass of RNA, essentially, right? That's what you want to know per particle. We're not quite there yet, but stay tuned, okay? Next question, are the cartridges single use? Yes, they're single use, and our customers love it because it means there's no cleaning in between samples. And it's really the cartridges that afford all of the ease of use factors, right? You can run complex samples that foul up the cartridge eventually and not have to clean it out. You don't have to clean the flow cell. You also get this no alignment, no calibration required as a virtue of these consumable cartridges. So yes, they're single use, and it's a great thing. Last question in the LNP. Thanks for all these good questions. How stable are LNPs during measurements with the ARC? Why are all the LNP measurements presented a power law distribution with no apparent peaks? Is that always the case when LNP size is measured with the ARC? So it's not the case. Some formulations do show peaks when measured by our electrical technique, the MRPS technique, right? It depends on how they're produced in our experience, right? Some formulations are made by different methods and yield different size distributions. That's why it's important to be quantifying them, right? The LNPs are stable as evidenced by our replicate measurements, as well as that volume dependence of brightness that we talked about, that I presented earlier, shows that the particles are intact during the measurement, right? We're getting a nice scaling of brightness to particle size. And that shows that we're indeed staining the internal particle, the internal payload of the particles, and that they're intact also. So hopefully that answers your question. I don't see any other questions in the chat. Oh, one more. No, that's it. Thank you. So thank you all for your attendance today, for your interest. Please follow up with us. You can visit our website or send us an email at info@. We'd love to hear from you and continue the conversation. So thank you very much.
Thank you very much, Dr. Freykin. Thank you to everybody. And we look forward to welcoming you to another webinar. We now close this webinar. Thank you very much. Goodbye.